Colour patterns in nature are adaptations that have evolved for the reason of communication. These signals share information such as "I'm worthy to procreate with," or can be a warning called AGRC signals. Many organisms converge upon the same colorations. This is identified as mimicry. The armoured catfish, Gordon-Manley, inhabiting areas of South America, are one such species that capitalizes upon the positive symbiotic relationship of similar aposematic colorations, representing both populations' poisonous spine. Professor Alexandrou investigated relationships which are called Müllerian mimicry in the scientific journal Competition and Phylogeny determine a community structure in Müllerian co-mimics. The first aim of the study was to determine whether the species both displayed Müllerian mimicry as a result of homologous or analogous traits. The study examined 425 populations of distinct species and developed the following phylogenetic tree. It shows the lineage's history, separating each species within it. It demonstrates that each species has most likely converged through selective pressure towards the same aposematic features. Since all coexisting unrelated species that are participating in these mimicry rings are allopatric, as denoted by the colour patches on the diagram, a vast different and distinct evolution and development has occurred, giving greater evidence to this being an homologous trait. The second aim of the study was to determine whether coexisting unrelated species that participated in these mimicry rings had different diets. Evidence for this comes from the competitive exclusion principles developed by Gregory Gorse that states two species that compete for the exact same resources cannot stably coexist. To investigate this hypothesis, the catfish participating in mimicry rings had their diets analysed using two measurements. Firstly, a qualitative measurement of the catfish morphology, in particular the shape of the snout. And secondly, a quantitative measurement of the stable isotope ratios of nitrogen and carbon in their muscle tissue. As shown in the following figures, a clear difference in size can be observed, and more importantly, snout size. The barbell sizes, commonly known as the whiskers, are also significantly different. It is concluded these structural differences in the fish show each species has a different trophic niche. The second measurement was gathered by collecting muscle tissue of the sample group, made of 18 different species, drying it and scientifically analysing it. The isotopic standards were then compared with the National Institute Standards of Technology, indicating a considerable difference in distinct species diets. This study was cited 29 times. One scientific article, being an updated study, was titled "Competition and the Evolution of Imperfect Mimicry." In the study, the authors identified how coexisting organisms, to minimize interspecies competition, diverge phenotypically, resulting in imperfect mimicry. This develops on catfish mimicry, giving greater evidence why such a morphological difference and range has occurred, resulting in the clear imperfect mimicry.